The cell theory came into focus in the mid to late 1800s with the work of Schwann and Schleiden. And pretty much at that time, everybody was convinced that living things were composed of cells. So, for example, take a single-celled organism like an amoeba. Nobody debated whether this was a living organism, but there was debate about what was happening inside cells to keep the cells alive. The state of understanding of chemistry at the time was rudimentary, and so it's understandable that lots of scientists appealed to different kinds of ideas to understand what was happening inside cells. One of those ideas was something called vitalism. Vitalism was the view that living things had a vital force that governed the chemistry of life, and scientists appealed to the vital force when trying to explain what was happening inside cells. Let's take an example. Here's our amoeba. We've got the membrane, we have the nucleus, and we've already learned that glucose can be an energy molecule, and the energy is stored, so it's potential energy stored in the chemical bonds of the molecule. And we've learned that proteins, in fact a team of proteins, will need to interact with the glucose to begin breaking the bonds and releasing the energy. Now here's where the vitalism came into play. Because scientists in the 1800s and early 1900s didn't really understand what was going on in cells, they imagined there to be a serious problem inside cells. For example, take a single glucose molecule, and here's our protein that must do the first chemical reaction on that. Well, suppose they're far apart. How are they ever going to find each other? After all, the size of a single protein, the size of a glucose molecule, would be very small relative to the size of the entire cell. So this is sort of like one of the problems the Vitalist was trying to solve. How could these two molecules find each other so that the important chemistry of life could happen? Well, there's where the vital force comes in. Perhaps there is some governing force that will help these molecules interact so that the needed chemistry can happen. So we'll think of it this way, that the vital force is in a sense a sort of a commanding force, that the cell parts don't have the wherewithal, they don't have the capacity to immediately find their targets. They're just kind of bumping around, and then they would never find each other, so you must have some kind of commanding force that will help the molecules interact in a timely fashion. So that was the Vitalist solution to the puzzle of the chemistry of life in the late 1800s, early 1900s. But then with time, new views began to emerge, and we'll call this the Materialist view. So in Materialism, that would be the view that living things were physical objects and the laws of physics and chemistry were sufficient to explain the chemistry of life. No vital force was needed to explain cellular processes. So now consider the obvious difference between this picture and this picture. Back to our case of how do the proteins find the glucose molecules? Well, here was the materialist answer to the vitalist challenge. What's happening is the cell is making lots of these proteins, and in fact there would be lots of glucose molecules, and if you have enough of each of them, then chances are they will randomly collide in a short period of time. So you see, you don't need any kind of intelligent vital force guiding molecules together if you have enough of each. And so now random collisions would be sufficient to explain how the chemistry of life is happening. You don't need a vital force. So instead of molecules needing to be commanded by some intelligent force, the idea is they're just bumping around randomly. Now, it is true that the molecules will have to have certain properties, but these are properties already sort of built into the molecules. For example, the protein has to be able to stick to its target molecule. So the protein will have to bind to the glucose, and that feature that the protein sticks to glucose is a feature of that particular protein. So it's that proteins have certain properties of themselves. That's what allows them to do the unique chemistry that's going on in the cytoplasm. So today, modern biologists have a materialist view of a cell. So cells are composed of molecules, and these proteins are very important kinds of molecules. They have different structures, and it means that their different structures give them unique special properties different from each other. For example, this protein here will stick to the glucose molecule. Glucose will stick to it, whereas glucose will not stick, for example, to this protein. This protein can bind the green and the red molecule, but this green protein will not bind that red or green molecule. So the different structures of proteins give them certain properties. They will be sticky, in a sense, to different kinds of molecules. And this means that random collisions can still allow chemistry to happen, because if you have enough proteins and you have enough of the target molecules, you'll get enough interactions in a short enough span of time so that the chemistry of life can occur.